taxonomy and biodiversity conservation circuit. She is also a senior lecturer in the biodiversity and conservation at School of Science in Monash University, Malaysia. She is the co-chair of International Union for Conservation of Nature Species Survival Commission Firefly Specialist Group that works to conserve threatened firefly species around the world. She also spoke at TED Women 2023 in Atlanta. She spoke about her personal and scientific journey exploring the mysteries of fireflies. Now, after all that, let me introduce, with no further delay, Dr. Wan Faridah Akmal. Help me welcome her. Give her a round of applause, please, Dr. Wan.
Okay, thank you very much, Ray. That's a very long introduction of me. It's good to be here. I think I'm not going to be on stage because I think you want to say what's on the slide. Next, please. Okay, so I think you need to click. Yes. When we talk about fireflies, we always look at this one. I'm not sure if you can see it. But it's always about synchronous flashing fireflies. So there's like what we call it clip. So everybody, when we talk about, I study fireflies, and they say, in Kuala Selangor. But this one is actually recorded by my student. She's not here, Nicholas. It was in Sabak Bernam, Selangor. So Sungai Bernam is a border between Perak and Selangor. So it's one of many wonderful areas that you can see fireflies. But, next, please. Okay. Alright. There are not just one type of fireflies. There are many types of fireflies. What you see is what just now it was synchronous flashing fireflies, what we call it pteroptyx. Pteroptyx as a species. But what you can see around here is there are many kinds of fireflies. The way they flash, the way they blink, the way they flicker, they are different. But look at that. You can also see there are spots there. There are whitish spots. 2 spots, 1 spot, that indicate different kind. One is female, one is male. There are also female that cannot fly at all, but they are looking at in the form of larva. Because this is an interesting thing from the perspective of science, because we want to see them, because why do they actually don't have even wing for the female? Because they want to save energy so that they can produce eggs, a lot more eggs, so that they can sustain the population in the future. But there are also the one that you look at that, a female with a short wing. So also another way of reducing energy so that they can focus on reproductive biology. So, and then you can see the male looks like adult beetle, right? And then you can see the larva glowing, different kind of larva indicate different kind of species. So that's how wonderful it is. Just looking at fireflies. They are just, flash pattern is different because it indicates uniqueness of each species. Okay, next please. Oh yeah, the star of the light show. So that is what I want to share just now, that you want to blink is the light organ, lantern. So in the world, I'm working with firefly experts around the world, and we are going to have an international firefly symposium in Mexico, Tlaxcala this year. So every 3 years, all of us together somewhere in Europe or somewhere in Asia. We choose the place. Now we have the number, more than 2,600 species have been recorded in the world. And how many actually in Malaysia? Next please. More than 60 species. Did you know North America alone has 200 species? Doesn't make sense, isn't it? Because we are in the biodiversity hotspot. We should have more. By saying we should have more, because it's a lack of studies, because we don't have many people. I am one of the 2 taxonomies in Southeast Asia and the Australia and Pacific region to actually study fireflies. We are lacking of people to study. We don't have many. It's not because we don't have many firefly species. It's just because we don't have many people to study yet. But we are also racing against time to discover them because of the development happening really fast compared to the way we actually discover or do research, isn't it? Okay, next. So, Leon already indicated just now, we have, at first it was discovered 7 and then there are 8. So what you see is there are all 8 up until January 9, 2025. I have the exact date. When we thought that there are only 8, so my students have like this all like been collecting and then looking at the specimen. You know, we have Stenocladius. Do you know how it looks like? So at night, Stenocladius has 28 light spots glow. So you only can see them glowing at night, but only when they appear as a specimen, this looks like a worm. If you found them in the daytime, they look like just worm, right? Diphanes, Pyrocelia, they produce green glow at night. But how do you indicate whether it's Pyrocelia or it's a Diphanes? It's something that you have to join our firefly walk. And then you can see how the flash pattern different from the other, from each other. And then we have dry luster. Dry luster usually can be only found or through a larva, not yet adult, because the adult choose to live somewhere very discreet, somewhere very hard, hidden somewhere else. And then we have Pygolucilla dungana. It was named after Dungon because it was part of an area that is discovered first or mainly in Dungon, Terengganu. And then the next one, next please. The star of this Bukit Kiara, we always know about Lamprigera. So the glide, there's a huge larva, glow about 7 centimeter. It can go to 10 centimeter, and then you can spot them very easily walking across walking on the tarmac road or forest trail. Next, and this one is the adult Lamprigera. Very rare can spot because you have to wait for him to come. So this adult male only appear right after sunset, only for 30 to 40 minutes. After that, they go rest and they're not coming. They will come back tomorrow. So this is very short window that you need to understand ecology. Why we only found this male quite late because we couldn't find where is the male. We only look at the larva. But the problem is, next one please, because it only have 30 to 50 minutes and this is the female. So the male is looking for this type of female that cannot fly. And imagine, it cannot fly, it doesn't have wings, it doesn't move, but it has very bright light so that the male can locate in 30 to 40 minutes. Very challenging, right? And then you have sky glow. You also have artificial light at night. How do they see each other when light is their communication? Okay, next one. Then this is the size of female and male. So 2 days ago, fresh from the oven. Now this is from the lab. My student Jack is actually working on the breeding of Lamprigera, looking at their life stage. So you look at it, it's what we call it, sexual dimorphism. Like if that is the male of Lamprigera, that is the female of Lamprigera. The size is huge. It's actually looks like totally different species, right? So imagine, look at the body, how they actually, the habitat must be preserved well. The humidity must be good in order to sustain their habitat, right? How they can survive. Next. So now this is the life cycle. We'll skip this part. It's just that how the process is happening in order for fireflies to actually survive in one population. Next, please. And this is what you see camouflage. If you are going to Clady Trail, if you can spot this, you have good eyes. This is actually larva of Pygolucilla dungana. Only they have spiky. They are not harmful, but they actually use that spike to anchor their body on the substrate so that they can still have some kind of water around there, but they are not flowing away or they're not going to be submerged too much in the water. Okay, next. This is what we prove. Direct observation, they eat snails. They are carnivorous. Okay? So you also have to save snails. Alright, next. This is Pyrocelia. Sometimes we call it ghost firefly because they will just like on and off the light at night, green glow. Next. This is how it looks like when they are actually walking, the larva, or sometimes mostly the larva walking, green glow. And this is Colophortia. So this is what we're going to get, the mystery of this spring. Oh, sorry, I've already folded this one. If you have the flyer, because this is what made us confused for over 8 years. So we always have seen this Colophortia. Next. It looks like the same. 8 species until 9 of January 2025. When? Next. Jack, I assigned him to see, Jack, can you sort the specimens? And then look at the Colophortia and tell me what's the difference. And then one day he said, Dr. Wan, I think we have a problem. So the problem is, something looks different because when you see under the microscope. Next. So, because there's only one specimen, I don't believe, not is that I don't trust him, but inside, as a scientist, you have to have more data in order for you to not say, oh, I mixed up the specimen, Dr. Wan. So you don't want that to happen, but that happens. So we actually look again for, go back to the coordinates where we actually collected this specimen. Next. And this is what my student photographed up close. Something for me as a taxonomist, when we look at it, there's a bipartite, that what we call it, the white spots actually have 3, looks like Colofotia, but there's something at the wing, and it's actually folded. You will get the chance what I mean by folded. Next, please. So, when it's folded, we look at it. I have been to Natural History Museum, London. I remember it was my mentor, Leslie Valentine. She told me, Wan, since you are in London now, can you check one pteroptyx from Gombak that I actually think is new to science. That was in 2015, based on collection 1921. How come our specimen in London? That's another story for another time. But then we have one specimen. And then later after that, she said, yes, it's new to science. Can you take pictures of for me? So that is the one, the folded wing, that give me a clue. The second one is because it has some long appendage. That the point here, you can see from the arrow, that is something that Colofotia doesn't have, and other pteroptyx, including kelip-kelip in Kuanpong, Kuantan, Selangor. Okay, so there was one second specimen was from University of Malaya, zoological specimen. One day I spoke with the curator. I said, Professor Sikumar, can I borrow your specimen? I want to see what we have. And it turns out we have second specimen, 1935. And then, this is the 2 specimen. And the 3rd specimen in the world, next, is actually similar to this one, which is in mangrove. All of these firefly species has the bent wing. They are all found in mangrove forests. So we were just like, clue, is this a mislabel? Until, next, we see this in Bukit Kiara. This is the first live sighting of pteroptyx gombakia, 3rd specimen in the world. But this is the only one that is found not based on museum specimen, but also confirm the sighting that this is the first species of bentwing that is not from mangrove forest. It's a new information for scientists as well. So with that, I think that's I end my slide. Thank you very much, and then we will see next for this one. Thank you.
Thank you so much, Dr. Wan, for that very interesting